Ekis, from the Llano Estacado to the Rio Grande Valley, from the bayou to the bottomlands, from the desert to the rolling plains. This is a seminar for a wide range. Coming to you today on the Soil and Crop Sciences Hour of Power, a brace of the department's finest, coming to you from the great student-producing region of College Station. Dr. Murray, would you like to introduce our first speaker?
Sure. It's my pleasure to introduce Shakirah. Shakirah is a native of Uganda. She got her master's at Makerere University in Uganda, working on perennial sorghum. She came to join my program at Texas A&M University and working with some other very exciting experts in sorghum perennialism like William Rooney and Russ Jessup and Dr. Muthu. And she is pushing the frontiers of what you can do with perennial sorghum through both UAVs, analysis of hydrogen cyanide, and biomass. So without further ado, Shakirah, one of the hardest working students in the Texas A&M University system.
Right. Thank you so much for your introduction, Dr. Murray. All right. I'm Shakirah Nakasagga, presenting my research on estimating hydrogen cyanide, biomass yield, and unoccupied area system-based regrowth in perennial sorghum and derived lines. A background to my study: sorghum is the fifth most important grain crop all over the globe, and the US is the world's leading producer with 9.27 million metric tons. And in the US, sorghum is the third most produced grain, mainly utilized for forage and for biofuel. Though there is a small percentage that is being utilized for food, since sorghum is gluten-free. Statistics show that Texas is the second leading producer, is the second largest producer of sorghum with over 62 million bushels after Kansas. Sorghum has two species. We have the perennial species and the annual species. For the perennial species, we have Sorghum halpens, also known as Johnson grass, and this is a tetraploid. We have Sorghum propinquum, which is a diploid, Sorghum almum, which is a tetraploid. All these are wild species, and they are characterized by rhizomes. Rhizomes are horizontal underground stems that store nutrients and can also be used for vegetative propagation. For the annual species, we have Sorghum bicolor, and this is the domesticated species commonly used as food in continents like Africa and Asia. A brief background to my research project: it is being funded by the Land Institute. The Land Institute is a non-profit organization based in Kansas, and this is involved in perennial agriculture with an aim to enhance environmental sustainability. Different perennial crops are being developed, such as perennial grains like wheat and sorghum. We have perennial legumes like chickpea, then perennial oil crops like silphium. I'm going to focus on perennial sorghum, and the materials that I'm working with were developed by Dr. Stanton Cox and Dr. Fiona, with a long-term goal of producing a perennial grain sorghum. However, right now, when these materials are still in the breeding pipeline, they are being evaluated to see if they have potential for forage and also for use as a bioenergy feedstock. Why am I advocating for perenniality? First of all, for this specific perennial sorghum, they've been shown to have the overwintering capacity, and also they quickly regenerate after a period of extreme weather conditions. in comparison to the sorghum that is sown. And also there are additional benefits ecologically, like an increase in the soil carbon and also a reduction in soil erosion.
Hey, Shakirah, can I suggest you switch to presenter view? You're showing the screen or you're showing the presenter view screen. If you can switch to the screen you're sharing, it might be easier for everybody to see.
Okay. All right. Sorry about that. Let me see. Okay. Let me first scan. I don't know why it's refusing.
Yeah, just go back to sharing your screen. You're sharing monitor 1, just share monitor 2.
Okay. All right. So can you see properly now?
Yep, that's a lot easier. Thank you.
All right. Thank you. Thank you for that. I didn't know. All right. So for the limitation of the materials that I'm working with, first of all, previous studies have shown the capability of these materials to produce high amounts of hydrogen cyanide, which is toxic to ruminants. However, since these materials that I'm working with have been crossed with the sorghum bicolor, there could be chances that they do not produce high amounts of hydrogen cyanide in comparison to the wild materials. And also, sorghum halpens, which is one of the parents of these materials, has been rated as among the top most invasive species. However, these materials, since they are a derivative of halpens and sorghum bicolor, literature has shown that they have 10 to 12% of the perenniality, meaning that they cannot be invasive. And also gaining a balance between the grain and the biomass yield, since originally these wild materials are very vegetative, is not that easy. However, selection for various types for either seed or vegetative growth is being done in order to address this issue. And also perennial breeding requires maintaining plots all year round, which is quite expensive. And then I will nail on the perennial evaluation since it is time consuming. Traditionally, the way perenniality is evaluated, it is done by counting the number of rhizome-derived shoots, taking the distance between the crown and the point of emergence of the rhizome-derived shoot, and also counting the number of rhizome buds. This is quite hectic, very tiring, because you have to do a destructive sample, and also it is then consuming. That's why in this research, we are trying to see if we employ the unmanned area system, if we can be able to cut down the time spent on evaluation. Therefore, the objectives of my study are to determine the hydrogen cyanide in sorghum bicolor and sorghum halpens, to estimate the biomass yield and composition, to use unoccupied area systems to predict earlier and more accurately which lines have the most early season regrowth and largest yield potential, and also in addition to differentiate structures that contribute to overwintering in Zea diploperennis, which is perennial maize. For the first study objective, which is to determine hydrogen cyanide, this was done in College Station here at the Texas A&M AgriLife Research Farm, and we evaluated 100 materials, and the largest proportion were a cross of sorghum bicolor with sorghum halpens. These were derived according to Figure 3 and they are a tetraploid population. These materials were planted in a randomized complete block design with 2 replications, having 1 row plots, and hydrogen cyanide was data on hydrogen cyanide was collected last year at maturity, this year at regrowth in the mid-spring, also at flowering stage in the midsummer, and also at the late summer when the materials were mature. The protocol followed for analyzing the hydrogen cyanide content was developed by Texas A&M Veterinary Medical Diagnostic Laboratory, and the steps that were followed were, first of all, we sampled 3 to 5 plants in the field. Then we got the fresh leaves. We weighed 300 of these leaves and digested in a flask using chloroform. This was incubated at 37 degrees Celsius for 1 hour. And we had a qualitative assay whereby we used indicator paper to rate the rate of color change, which was associated with the hydrogen cyanide, whereby 0 meant there is no hydrogen cyanide, M meant very low hydrogen cyanide, 2 meant low hydrogen cyanide, 3 meant moderate amount of hydrogen cyanide, and 4 meant high amounts of hydrogen cyanide. In order to get more quantitative results and also to do some downline applications, like running the analysis of variance, we included calorimetry and used a calorimeter which gave the L, the A, and the B values. The quantified color change was obtained using this equation, whereby according to this color solid, L means lightness, A means green and red vector, and B means the yellow and the blue vector. In order to validate the methodology used, we built a hydrogen cyanide curve, which was gotten after varying the amygdalin concentration. Amygdalin is the manufactured form of hydrogen cyanide, and it is made from bitter almonds. To this amygdalin concentration, an enzyme known as beta glucosidase was added. And as you can see in this figure below, 0 amounts of amygdalin, there was no color change. And as we increased the amount of amygdalin, the color changed, becoming more and more red. And the data that was obtained from this study was processed in GMP. The hydrogen cyanide curve is portrayed in figure 7 here. And you can see as we increase the amygdalin concentration, also the color change increased. So the results from this first study, both the calorimetry and the qualitative analysis, first of all, for the pedigrees that were assessed last year at maturity, there was no significant differences between them, meaning that they were consistent. And also for the qualitative analysis, most of the plots had low amounts of hydrogen cyanide as according to the qualitative scale. This year at regrowth, however, the pedigree were significant. And as you can see from the color distribution in the qualitative scale here, most of the materials had low amounts of hydrogen cyanide, but still we had some of these plots, these are plots, we had some of these plots produce moderate amounts of hydrogen cyanide and also high amounts of hydrogen cyanide, meaning that there was no consistency. Some of these materials produced high, others low, and others moderate. Then during flowering in the midsummer, the pedigree was not significant for the hydrogen cyanide, and you can see that most of the plots now increased, those that did not produce hydrogen cyanide. And at maturity, still the pedigree was not significant, and the number of the plots that did not produce hydrogen cyanide further increased. Therefore, in order to see the differences between the growth stages, an analysis of variance was run for the different growth stages, and stage was significant. And you can see this in figure 8 here, whereby at regrowth, most of the materials produce hydrogen cyanide, but this reduced at flowering and it further reduced at maturity. When a combined analysis of variance was run at maturity for last year and this year, the year was significant. And you can also see this in this figure here, whereby last year, most of these materials produced higher amounts of hydrogen cyanide compared to this year. And this can be related to the weather conditions that at the time I took the data, the temperatures were higher in comparison to this year, the time I took the data, even though it was the same stage at maturity. For the second study, it was to estimate biomass yield and composition. Biomass yield was obtained per plot basis, and we got both the fresh and dry matter yields. For the biomass composition, this was based on dry matter yield, and we used near-infrared spectroscopy in order to analyze the biomass for the different components, such as cellulose, hemicellulose, lignin, among others. The results portrayed here are for last year, and for the biomass yield, the pedigrees were significantly different for both the fresh and dry matter yield. And when a comparison was done between the grain sorghum and the perennial sorghum, for both the fresh and dry matter weights, you can see that the perennial sorghum had double the amount of yield in comparison to the grain sorghum. And for the biomass composition, the biomass had 30% hemicellulose, 26% cellulose, 13% lignin, and these constitute the structural carbohydrates that in total were 69% in comparison to other components, even though there was a proportionate amount of soluble carbohydrates, which was 12%, ash content, starch, and protein content. When the analysis of variance was run for the different biomass components, you can see that the pedigree was significant for the cellulose, the hemicellulose, and the starch. When a comparison was made between the perennial and the grain sorghum, for the grain sorghum, it had significantly higher amount of ash and protein content, while the perennial sorghum had significantly higher soluble carbohydrates. For the third study, it was to evaluate tree growth and biomass yield using unoccupied area systems. We used a rotary wing with an RGB camera at high resolutions. Images were taken for last year and also for this year of the sorghum field. So the data processing, first of all, we also mosaic the images using a Geosoft Metashop. I created the shape files and cropped images in QGIS, and this is the step I'm on right now. I'm processing for the vegetation indices in R. And after this, I'll be making regression between those vegetation indices with the biomass yield that was obtained using the combined harvester and also the perennial traits. For the perennial traits that I'll consider, there'll be the number of the rhizome-derived shoots at regrowth and at maturity, and also the number of shoots from the crown, since these are above ground mass and can be captured by the UAV. For the fourth study, it's like an additional study, and it is to do with the perennial maize, which is Zea diploperennis, and basically it is to characterize overwintering in this material. As much as in sorghum, it has been confirmed that it is the rhizomes that contribute to perenniality. In maize, it is questionable because the regrowth can be seen not only from the rhizome, but also it can be seen from the crown, meaning that there could be other factors that contribute to perenniality in perennial maize. So the materials that were used for this study, there were 5 Zea diploperennis lines, and these were planted in a randomized complete block design. First of all, we analyzed the composition of the underground structures, and this was done by, first of all, having a destructive sample, and then we separated the rhizomes from the crown. These were oven drained and ground and scanned using an NIR. This was the spectrum that was produced, and we used the spectrum in combination with a rhizome curve that was developed by Washburn in order to make predictions of the different components within the underground structures. So the results showed that the pedigree, they were significant for the crude protein, the starch, the water-soluble carbohydrates, and the crude fat. And for the underground structures, that is the rhizome and the crown, they were significant for the crude protein, the starch, and the crude fat. When we made a comparison between the rhizome and the crown, you can see that the crown had numerically higher amounts of all the components, meaning that there is something that contributes to overwintering, not only the rhizome in perennial maize, most especially when you look at the composition. So we concluded from this study, first of all, in the first study, the perennial sorghum produces higher amounts of hydrogen cyanide at earlier growth stages, and therefore it should be given sparingly to the animals, or better to be ensiled. And also, having included the calorimetry, it showed that it is a potential tool for color quantification of the hydrogen cyanide qualitative assay, and this gives opportunity for data manipulation and also to increase the objectivity of the study. For the second study, which was about biomass yield and composition, the perennial sorghum showed that it is a potential source of forage and bioenergy considering the high yields. And also the biomass had numerically higher structural and significant soluble carbohydrates, meaning that it is a potential bioenergy feedstock. However, the limitation to the second study, we did not include forage and high biomass sorghum in order to have better comparisons. For the third study, it is still in process and no results and no conclusions have been made yet. For the fourth study, the underground structures were significant for the crude protein, the starch, and the crude fat, and all these components are essential for overwintering. However, there is need for further differentiation since we've not yet gotten to the real conclusion of this study. We've not yet made definitive conclusions about, since it is still ongoing. And then the crown accumulates numerically more assimilates than the rhizome. This could be a potential reason for overwintering and regrowth from the crown in perennial maize. However, still research is still ongoing. I'd like to take this opportunity to thank my committee for their support, to thank the to thank the Texas Veterinary Medical Diagnostic Laboratory for the input, the Quantitative Maize Breeding Program, and the Sorghum Improvement Program for their support and input. I'd also like to thank the Land Institute and Mr. Bob Hatch for the funding, Texas A&M University for the admission, Texas A&M AgriLife Research for the resources, and thank you all for listening.
Great job, Shakirah. Very nice. Questions?
Shakirah, this is Wayne Smith.
Yes.
Tell us a little bit more about your pedigrees. I missed the first couple of slides. Sorry, I was a little late. So tell us a little bit more about your pedigrees.
All right, so the materials that I'm working with, I'm having most of them are a cross of sorghum bicolor with sorghum halpens. Then I have some materials which are a cross of sorghum bicolor with sorghum propinquium. Then some pure lines for sorghum halpens. I have one line, and also grain sorghum, which were gotten from Dr. Rooney's program. And for the materials, most of these materials are a tetraploid population that were developed according to this scheme. The sorghum bicolor was doubled.
So you sift and then you back cross to the tetraploid, and so all of your 80 lines are full sieves. They all have the same parental pedigree.
Yes.
Okay, that's what I was wondering about. Thank you.
You're welcome.
Other questions? Step right up. Don't be afraid.
Hey, Shakirah, can y'all hear me?
Yes.
Okay, my name's Jenny Dudak, and I actually have a question. So about the HCN levels. So do you think there's a way that, like, by crossing, you could reduce the levels at regrowth? I mean, are people working on that? Is that something you think is worth time?
First of all, like naturally, even the sorghum bicolor, all sorghum naturally produces all sorghum naturally produces hydrogen cyanide. But it's just that for the wild types, they've been shown to produce more hydrogen cyanide at specific points of growth, like at emergence and also like at regrowth. They produce more amount of hydrogen cyanide. But yes, even though like all sorghum produces hydrogen cyanide, the rate at which the sorghum bicolor produces is less in comparison to the perennial sorghum. So that's why I took this study, because it is a cross between the wild and the domesticated to see how much, but still at this, they are just specific growth points whereby they will produce high amounts. It's kind of difficult, and I cannot confirm to say that it can be eliminated. It can be eliminated completely, because naturally it does produce this durin that is broken down at specific points and even at extreme weather conditions to produce hydrogen cyanide.
Thank you.
You're welcome.
All right, we have time for one more question, if it's a short one.
So I'll throw in a short one, Scott.
Right.
Your second year of data, did you reestablish the plants, or was that the second year of growth?
This is the regrowth. It's the second year of growth.
Okay, thank you.
You're welcome.
Thanks again, Shakirah. Very nice presentation.